Thank you, organizers. I'd like to inform everyone that I'll be using two devices. One input for my PowerPoint and my video, and the other is using my other device for my audio. So um, for this session or for this talk, this may be a bit different, but also related to what has been discussed earlier. We'd like to share with everyone how we were able to deliver a laboratory and field-based course during state of emergencies, like what is happening right now, and how we have able to teach ecology using OERs or open educational resources amidst COVID-19 pandemic in the Philippines. So together, on behalf of my co-authors who are with me right now, I'd like to present and share with you our experience. And as you know it, perhaps I do not need to define ecology, but it is actually highly related and very relevant to what is happening right now. So the department, even the university, has pushed through uh, in offering this course, regardless whether this is online or regular face-to-face course. And we acknowledge the recent paper of Montgomery and McDonald, how they were able to highlight the interconnectedness of the world upon realization and by highlighting the origins of different emerging diseases. And one is COVID-19. And the unprecedented interconnectedness of global society has now created a new balance of pros and cons. And as we acknowledge, as global citizens, it is in our shared interest then to preserve human health and conserve biodiversity and natural world. So it is a good call now for creating a more sustainable future for people and animals. So I begin my talk for today with this paper because we want to make sure that um, our purpose to offer ecology, lecture and laboratory in the university in the Philippines has reason because we want to make sure that by teaching ecology and allowing the students to learn ecology despite the situation, they would be able to prepare for future challenges, public health challenges or crisis with such course. Okay, so this course is being offered, to be specific, Bio 426 Laboratory is a 2-unit laboratory course being offered by the Department of Biological Sciences in the College of Science from UST. UST is the oldest university in Asia, just a side note. And this is being taken by the students of BS Biology program. Most, if not all of them, are second year level. And this is usually and regularly offered during first term of academic year. And BS Bio majors in the university has 3 tracks: medical biology majors with 7 sections, environmental biology majors with 1 section, and industrial biology major with also 1 section. And this course has been received by a total of 341 students, which is the largest BS biology student population in the Philippines. And just to give you an information what we really intend to teach, whether it is regular face-to-face or even virtual enriched mode, the Bio 426 laboratory should be able to guide students in the process of research in ecology. And by this, they should be able to be introduced and they should also practice basic ecological methods and with focus that on the particular areas of ecology. They should be able to deal with basic laboratory and field exercises related to the ecology of climate or the environment itself, different types of ecosystem like terrestrial ecosystem and aquatic ecosystem, even the population or the species, individual species to be found in such environment and how they get to interact within their own population and how this population should be able to or are being able to interact with other types of population or a group of population within a particular ecosystem or environment. So with this focus and rationale, we should be able to picture out already the possible challenge or challenges that any facilitators should face if we are to offer this virtually. with the outcome that we expect the students, once they have completed this course, to know how to sample, process, analyze the data at different ecological hierarchical structures, like what I have emphasized earlier. So in a regular setup, we offer this course, of course, in the campus. So this is a picture of the main building where the laboratories and lecture classrooms are found. Also, there's a satellite or a different building for different laboratories. But basically, the College of Science is housed in this building. We call this main building. And this is the regular picture that you get to expect if we will offer this in a regular meeting or face-to-face meeting. This is how we look, how we conduct laboratory classes. Even these exercises can be applied in the field, like what you can see here. And most of our time, or most of the students' efforts, should be devoted to apply what they have learned in the lecture, and even in the laboratory, they should be able to apply this in the field. So every offering of this semester, of this course, they should be able to apply the ecological methods, the learning they have, to field collections and field processing, and even analyzing the data they have obtained from the field. However, with the present situation, we cannot do this. And we are only limited with enriched virtual mode of learning or modality. As such, we made use of what we have been using also as a learning management system, like Blackboard. In terms of our synchronous classes, we've used Zoom. And if they need to communicate with us, use some Google applications, then we have also, the university has subscribed to G Suite accounts. And with this, uh, basically Blackboard is not really new for the university. This has been the LMS of the university since 2002. Even before the COVID-19 pandemic, submission of assignments, like platform for short quizzes and formative exercises, even deployment of some learning materials, are being already used or submitted through Blackboard, or what we call now UST Cloud Campus. However, this is the only time that we will use Blackboard back then. But now, it requires us to offer, fully deliver, Bio 426 Laboratory, this entire course outline, with the use of Blackboard and other learning materials, or learning modalities rather. So imagine we have this number of exercises. We actually reduce it already, significantly reduces, so that the students can cope up and be able to still get the essential concepts that we intend to share with them for Bio 426 Laboratory for the duration of 18 weeks. So in the university, our calendar is divided into two periods. That is prelims and finals period, like any other university, I suppose. And prelims, or the prelim exercises should allow the students understand the environment itself by being able to pinpoint some sampling sites, by being able to map their species distribution, a particular species distribution, or even their sampling sites. So that will be covered by the first exercise. And the succeeding exercise, if you will notice, this will talk about, and this will capacitate the students in assessing and understanding the environment, from the climate to specific parameters from aquatic and terrestrial environments. Once the finals period comes in, this time the students are now prepared to understand what are the organisms, or to understand the organisms they need to see in those environments that they were able to assess. So this time they should be able to know how to sample aquatic organisms or terrestrial organisms, even the environment itself. And here we also focus on different hierarchical structures already, which is in terms of population and community ecology. So those are specific topics already that must be covered by the students. So this entire course outline should be delivered. So that's actually the challenge that we face prior to offering this course. So how we'll be able to successfully deliver this laboratory and field-based course using an enriched virtual mode. So that's pretty much the challenge that we had to, face and resolve. So for this session, I'd like to share with you how we were able to fully deliver Bio 426 laboratory through enriched virtual mode, and how we were able to utilize open educational resources, or OERS, in covering Bio 426 laboratory learning outcomes. So our approach in our every meeting in the laboratory is divided into this two class flow. So basically, each class will be, or we meet each class twice a week. Each meeting has 3 hours, contact hours. But of course, that will be tiring, of course, for the students, and might be, if not efficient, if we will always meet them every week with within 6 hours, contact hours. So we divided into two. So the day one should be devoted for synchronous classes. Here we should be able to do semi-synchronous, like giving them pre-tests without being, online with us, or they should be able to watch the recorded pre-lab. That has been the practice already. We record our pre-lab instructions and informations before they conduct experiments, so that this may be accessed by any students regardless of the status of their internet connectivity. So at their own pace, they can prepare for the laboratory exercises already. Now, the half of this day one should now be devoted for fully synchronous classes or pre-lab discussion. Here, we should hear student clarifications or questions from the students. We can give them recitation or activities that we think are crucial for the exercise. And the other half of the delivery is devoted for asynchronous class during day two. So here, we either give them the remaining 3 hours to continue working on their exercises or lab activities. We can also give them post-lab quizzes or summative exams, or even it's their chance to submit their outputs. So this is pretty much how their week would go every time they have ecology laboratory course. So this is the typical, at least some parts of our course site using Blackboard, where the students can access all the exercise pre-lab videos and PowerPoints or PDF copies of the PowerPoint presentations. And then they can also access the different handouts aside from the recorded pre-labs. Here, this is a more detailed handouts where they can refer to understand further the laboratory exercise or activity. So if you notice, how now did we deliver the activities? How did the students receive the OERs or other experiments, home or virtual experiments? So we have three teaching and learning strategies employed. One is the use of, or one is software utilization. The other is virtual and home experiments. And lastly, we use or employed modeling and simulations. For the first teaching strategy, these are of course subscription-free location mapping software application like Maps.me. That they can all have this in their smartphones. They can access a GPS type of application, and they can locate sites or potential sampling sites. And they also have the option to download either QGIS or Diva GIS, depending on the computer specs or hardware that they have. So that much, we are really being flexible so that the students can really cope up with the required software that we hope them to use. So these are just some of the examples or sample GIS mapping outputs from our students. And we are very happy that these are being received well and being accomplished well by the students. Imagine we have 341 students, but of course, some of these outputs are of course grouped by groups of 4 or 3. But we at least know and see how the students get to develop this type of skills regardless of teaching modalities. We also allow the students to use Canopy software, where they can also use this another subscription-free Canopy analysis software. This time, of course, they did not collect the pictures. Instead, we give them the pictures already. The only thing that they need to learn now is to use how to use the software. Because I think that's something that is very important, at least for them to learn at this point of time. So they were able to analyze the data they were able to collect. Okay. And also for the virtual and home experiments, this time they were able to do some ecological methods analysis in the laboratory that is usually done in the laboratory, but this time it's done virtually. All the materials, all the necessary steps or procedure are stated and shown, indicated in this virtual simulation. Aside from our supplementary videos, our personal recordings when we were conducting this in the laboratory, or even myself recording inside my unit, apartment unit, showing them how to use this type of equipment. So the students were able to remember and at least familiarize themselves on how to do this without them really doing it by their hands, by using some infographic poster, using the flow chart or schematic diagram like what you have on screen. And also, the students have enjoyed the use of home experiments like this. They were able to characterize soil type or soil classification using Mason-Jar soil tests, and they were able to analyze the soil type or soil classification for their samples. Of course, these are just collected at their backyards or any soil samples within their area. And also another thing that I think the students have really enjoyed based on our interviews and focus group discussion is this: the Winogradsky column, which they were able to monitor microcosm or microbial ecosystem. So imagine from day 0 to about 7 weeks or 8 weeks of observation, the students were able to monitor and observe actual succession or microbial succession based on the colors without using the microscope. But of course, they can already anticipate or assume at least the type of bacteria that they should be able to see in each color gradient for each sample. So these are just two setups. The one is on the left is the light setup and the other is the dark setup. And lastly, the teaching strategies we've also used are the modeling and simulation, like this simulations. When they need to check for carbon, of course, I'll be highlighting here the OERs that we were able to use from Annenberg and Lerner, from Virtual Biology Laboratory, and sometimes we also inject it with e-learning games, like Professor Paul has emphasized earlier. So here, the students were able to not just simulate and model some of the important ecological concepts, but they were also able to play it with learning, play with those simulations or those concepts while learning. So all of these street teaching strategies, of course, have to be assessed. So we assess them, like what I've mentioned earlier, we assess them by group. Most of our laboratory activities are accomplished by groups of 4 or 3. And of course, we also need to see how they have performed individually. So we
Excuse me,
provide.
excuse me, excuse me. Sorry to interrupt. But there is only 1 minute left, and you won't have any question and answer session. You only have 1 minute left.
Yep,
Yes.
I can wrap it up. Thank you.
Okay. Thank you.
So these are the feedback of the students. So from their expectation to experience to the challenges and suggestions. And basically the students, like what I've mentioned, have received all the learning materials and the OERs and the teaching strategies were found to be specifically the home experiments and the OERs were successfully delivered. And the home experiments were able to be um, one of the students' favorite strategies. Okay? And the bigger picture is this. I'd like to conclude my presentation with this bigger picture that OERs and innovative creative teaching strategies may provide quality education both in state and private colleges or universities. And as future directions, these are our future plans for the project or for the study. And these are the access to the OERs that we were able to use for your references. And I'd like to acknowledge the following facilities: thank you for your kind attention and for the
Yes. Thank you. Thank you also for your interesting presentation.